Good afternoon, everyone. Is my audio clear?
Yes, sir, it's clear.
Thank you. So in today's class, we will discuss some things about principles of molecular techniques. Um, there are some basic techniques that we all have to learn, and it's also a part of syllabus. And I think they are important for our experiments as well. So first thing is electrophoresis that we already have discussed when we were talking about DNA. And there are two more things that will be as a part of our experiments in the practicals lab. So these are the three experiments that we will do as in the lab. But otherwise, we are supposed to know these three principles that are involved with DNA. So electrophoresis I'm not going to discuss, but I hope you remember that it is electrophoretic mobility. We have a positive electrode and a negative electrode, and then all nucleic acids, or they have a net negative charge because of the phosphate backbone. And here is somewhere the well, that is where we load the gel DNA sample, and then it will travel. DNA will travel through to the opposite end. We will see the setup and everything when you come to the lab, but otherwise I think you have the basic idea. So electrophoretic mobility is based on the size. There is also one more thing we have discussed in the previous class about I'll just put it as supercoiling, right? And the concentration of the gel, with how much concentration. If you put higher concentration, then the mobility will be reduced. So higher the size, reduced mobility. Higher supercoiling, increased mobility. Higher concentration of the gel, then you will have reduced mobility, and so on. So simply what I want you to remember is when you have a mixture of DNA in your sample, and then you allow it to go through move in electrophoresis, then what you will find is that they form bands based on, if you have 4 different types of DNA molecules based on sizes, then it will form 4 bands. The one that is runs farthest is the smallest one, right? The one that runs the slowest is the largest one. That is simply what you should remember about electrophoresis. And today we will learn about restriction digestion, then we will learn about southern blotting and a little bit about northern blotting, and the focus will be also on PCR, polymerase chain reaction. And these are the two things that you need to focus right now in this class. So try to remember, try to pay attention. The first thing is we will learn about restriction digestion. When you say restriction digestion, I think the typical simple way of saying it is restriction enzymes. And we don't have to go through the terminology, the origin of terminology, and the history and all. We have a different course in your 7th semester that is about rDNA in medicine, recombinant DNA technology in medicine. There I take that course, I offer that course, and I discuss about real genetic engineering and other applied parts of whatever you're learning now. But it's an elective. But otherwise, in this time, if you want to follow through molecular biology, any of these papers, then you need to understand basic concepts, and that is the reason why these topics were included in your syllabus, because there is no genetic engineering course for you other than as an optional in 7th semester. So there are something called as restriction enzymes. You can also call them as restriction endonucleases. When you say endonuclease, I hope you remember that what is an endonuclease and what is an exonuclease, right? You have say for example, this is a double-stranded DNA, and if an enzyme is breaking the bonds from the ends, then you would call it as exo. Exo is outside or edges, you can say. And if there is a different enzyme that is trying to break up the DNA here, something like this, that is, it's making cuts inside, that is called as endo. So there are particular some enzymes called as restriction endonucleases, which are, there are two properties that are there. The properties are sequence specificity. Let me try out. So they are sequence specific. We will see in the coming slides. Sequence specific is one thing, and they are endonucleases. Okay. Exonucleases are not sequence specific. They are specific to the ends of DNA. If there is a free ends of DNA like this, then they just go and digest it from here on, so on. But endonucleases, majority of them are sequence specific. That means they do not go and digest break the DNA anywhere in general randomly. They would break the DNA only in special, when there is a special sequence. Those are referred to as restriction endonucleases. In what we know so far, in majority of the genetic engineering experiments, the type 2 are used. You don't have to know those things right now, but just for the sake, you should know that. Okay, I'm sorry, 1 second. There was a crash, I think. Okay. So here we are. There are nomenclatures. The enzyme, how do you name them? This also, I don't need that you have to mug up. But BAMH1, say for example, H, and this is usually the Roman number 1. Here it is, BAMH1. So the genus is Bacillus amyloacquisitions. So B is coming here, and this is the genus name. The first letter of the genus name is taken, and then the 2 letters of the species name, AM, is here, and then strain H, that is this part, and this is the first enzyme first restriction endonuclease identified from this organism. So you call it as BAMH1. Okay. Just remember, some of the things we will get to is some EcoR1, BAMH1, HIN3, and so on. I don't need that you memorize those two. But what you should remember is they have a sequence specificity as shown here. BAMH1 recognizes a sequence like this. So if there is a DNA that is having this sequence in middle, something like this, then BAMH1 is going to act upon it, and it is going to cut the DNA like this. So it is a sequence specific restriction endonuclease. There are many enzymes. As I was telling, there are actually about thousands of enzymes. If you go to the internet, you can find that, and each of them has some unique sequence, or their source is different, or the sequence specificity is different. Some of the ones, EcoR1 and BAMH1, are very common that people at least try to remember. And EcoR1 recognizes GAA TTC, whereas BAMH1 recognizes GGA TCC. I will get to what is meant by 5' sticky end and 3' sticky end, and so on. So number of bases it is recognizing is 6. 1, 2, 3, 4, 5, 6. Okay. There is something about the sequence, if you look at it, is called as a palindrome. What is a palindrome? You can read this sequence that is here. Here, you can read this GAA TTC. Read the same sequence on the, always reading should be done from 5 prime to 3 prime, right? So if you read the complementary strand, again you are getting GAA TTC. In this direction, GAA TTC, and on the complementary strand also we are getting GAA TTC. That is called as a palindrome. Either way you read, you get the same meaning. It is something like Abel was I ere I saw Elba. Again, if you were going to read from here, that's not exactly what we are doing with DNA, but just the literature. If you see EBL, say ABLE, here ABLE was WAS, you see WAS if you're reading from the reverse side. So those are called as palindromes. So there is all the restriction endonucleases, majority of them, if not all, when they recognize, their recognition sequence is always these kind of palindromes: GAA, TTC, GAA, TTC. You can also look it up with BAMH1 and many other things. Say, for example, I write GAA, TCC, GGA, TCC, right? Say I will write GGA, TCC, and this would be 5 prime, and here would be 3 prime. And if I have to write the complementary strand, GGA, TCC, right? They're all palindromes. So if I want to remember just eco R1, I just have to remember these three, right? If I know GAA, what would be the rest of it? TTC. If I know GGA, what will be the rest of it? TCC. You don't need to memorize. If I am giving you a question, I will provide you this information. I want you to rather understand the principles rather than mugging up. Okay. So here is one another way. There is an axis, as I was telling the palindrome. There is always an axis. As I said just a little while, just now, GAA, and the remaining would be TTC, and then the complementary would be GGA, TTC. There is an axis, right? It eco. This is the site for eco R1, and it recognizes a 6 bases, and there is an axis of symmetry like this. In general representation, they put something like this. The slash indicates where the endonuclease is going to break the phosphodiester bond. Okay, you know that you will have G, A, T, T, C. In all the cases, you will have, let me finish this, G, A, T, T, and C. In all the cases, you have phosphodiester bond joining these two, isn't it? The bases are inside, and the phosphodiester bond is something like this. So when eco R1 or restriction endonucleases or eco R1 break it, they actually break this part and here and here. That is where these slashes are indicated. So now you will actually have a scenario where you will have this part of it is one DNA molecule, and this one is one DNA molecule. If I have to cut it and show you separately, the DNA molecule would be, that is one DNA molecule, and the other DNA molecule would be something like this. Okay, so in the, if you zoom it out, or you see as a just a DNA molecule like this, if there is a site here, that is GAA TTC is here, then it would have been cut off by the restriction endonuclease like this. And now I also want you to remember or recollect what is this site. We are always writing 5 prime to 3 prime, and this would be 3 prime, and here is 5 prime. What kind of overhangs are formed? If I were able to separate these two, let me see if I can try doing that. No. Okay. In the next slide, anyway, we have a picture for it so you can understand. I hope you can see it. So here is a site for EcoR1, right? And restriction is happening in this place. Breakage would happen, phosphorested bond, and here also there will be a breakage. And then what you'll be left with two DNA molecules. This was one single double-stranded DNA, right? But because there is a breakage, you have, this is one part of it, and this is the second part. You have two double-stranded DNA molecules, but you have overhangs. This is called as overhang, right? Because they do not have pairing, or the complementary strand part is missing here. You can also call them as sticky ends, or you call them as overhangs. Okay. So what kind of overhangs are there? The 5' end is here, so you call them as 5' overhangs, or you call them as 5' sticky ends. But that is not the only way, only type of overhangs that are produced. Here we have an enzyme called PST. PST cuts in a different location. It recognizes CTG, CAG sequence, and it when it cuts, it's now leaving a 3' overhang, like in here and here. I hope you can compare what is the difference between 5' overhang and 3' overhang. Okay. But sometimes they cut, some of the enzymes like SMA1, they cut exactly in the axis of symmetry, because of which they form blunt ends like this. Okay, there are no overhangs, and no 3' overhang or 5' overhang. They have cut exactly in the middle, and those are called as blunt ends. So there are many enzymes, as I said, and some of them recognize 4 base cutters, some of them recognize 6 bases, and some of them are 8 bases. EcoR1 and BAMH1 that we have discussed are 6 base cutters. Why are we calling them 6 base cutters? GAA, TTC. How many bases is it recognizing? It is recognizing 6 bases. So you call them as 6 base cutters from here to here. I also want you to know that, say if I gave you, if I invert this sequence, here is the sequence, this will be 5', and this is 3', this is 5', and here is 3'. If I invert it, what can we expect is a question, right? Will we be able to, I can't know if we can do that. One second, let me try. One second. Okay, I'm inverting it. And what is it forming? Okay, the alphabets are inverted, but still you can notice that the reading frame has not changed. You will see G, A, A, T, T, C. Either way, it will come normal. So there is a difference, however, between, if I write C, T, T, A, A, G, this is 3', or sorry, this side is 5', and this side is 3'. The sequence 5' GAA TTC. I have given you two sequences here and here. These two are two different sequences, isn't it? One is reading, always reading should happen 5' to 3', GAA TTC. In the lower sequence, it is CCT, CTT, AAG. So keep that in mind because you have it. And say why I'm saying that is, if I have to write here the sequence TTC, right? It resembles the sequence of EcoR1, like I shown here. But this is a different sequence because reading should happen 5 prime to 3 prime. Okay, so there are several restriction enzymes that are used in to manipulate DNA, to cut the DNA, because the genomes, majority of the genomes, as we'll study in the later classes, they are very huge. One will not be able to use them directly for the purposes, because there will be it's difficult to find out if a particular sequence is present or not, right? And also, if I want to make any recombinant DNA technology, recombinant DNA, right? I want to clone a gene into a plasmid or a vector, then I should be able to cut the DNA, piece of DNA, and be able to ligate it. For all those purposes, gene cloning, or even for analytical purpose that we will discuss in a short while about southern blotting, we need to fragment the DNA, and we cannot use exonucleases because the whole DNA will be digested. So we have to use precise enzymes to cut properly, and that precision and control will be given by restriction endonucleases, because they are endonucleases and they are sequence specific. Those two are important parts of it. Now we'll look at blotting technique, which is an extension of why, at least in one way, why restriction endonucleases are useful. Uh, the point is that just please 1 second, be online. Sorry for the interruption. Yeah. So there is something called as a technique called southern blotting. It is after the scientist named Southern, and I hope you know what is meant by blotting. Say if we spill some water or something on the floor, we use some paper or something, sponge for example, to suck all this water, and so that we can clean it. That is something like blotting. Here the same kind of thing is used, where we are trying to take DNA from the gel onto a paper. That is the reason why we have the term blot in this technique. Southern is after the scientist. So the objective, for all these techniques, if when you are reading about techniques, it's always important to learn two things. One is objective. Why are we doing it? What does it say to us? That is what we should know. And yes, we should know the principle and then applications. Okay. And objective is something that I think is what gives the meaning to the rest of the things. If I remember the objective, I know the rest of the things. In southern blotting, the objective of southern blotting is to find if a specific sequence, if a sequence is present in the DNA sample or not. Present in the DNA sample. When you are analyzing um, DNA, then it is southern blotting. Okay. So the objective is to find if a specific sequence is present in the DNA sample or not. Okay. DNA is long, and we don't know if a stretch of say 100 bases is present or not. To do that, we use this technique. The first step is to, because the DNA is like this, and there are so many cells, and so many cells have so many chromosomes, and then they are diploid. So you get an entangled mass of cells. And in that, I want to know if a particular small if a gene is present or not. For that, we need to, for ease of doing things, we cut the DNA into smaller fragments and do electrophoresis. Okay. The first step would be restriction digestion. And after restriction digestion, we do electrophoresis. And once you have electrophoresis, DNA fragments will separate on the gel, something like this, if you can see. They will be, they will separate something like this. The bands will be there. Multiple bands will be there if you're working with genomic DNA. And after that, we will do the blotting. What is blotting? As I said, now the DNA is present in the gel. From the gel, we need to get the gel to the to a nitrocellulose filter or membrane. If I want to do that, I put the gel here, okay, as it's shown in the picture as a plane, and on top of it, I will put the nitrocellulose membrane here, and then I will add some paper towels and so on. And in this tank, we pour buffer, and that buffer is, there is, these papers are put, which acts like a wick. It picks up the buffer, and it will travel through capillary action, will travel through the paper towels here. What is it doing while it is moving from the gel to the top? It is carrying the DNA that is present in the gel here towards the upside, towards the top. When it is doing that, there is something that will happen. The nitrocellulose filter has high affinity for DNA. So while the buffer is moving, all the DNA gets caught onto the nitrocellulose filter as it is put here. This is the membrane. That is where the DNA will get attached, right? We are blotting the DNA that is present in the gel onto the filter using capillary action. Okay. And once that happens, we separate the nitrocellulose filter onto which the DNA is bound now, and then we will add a probe. What is a probe in this case? A probe is something, it is a nucleotide sequence. Say, for example, I want to find out, for ease, I'm giving you a small sequence. Usually the probe can be larger. So I want to know if there is a sequence like this, right? My objective, I'm restating it. I want to know if there is a sequence like this in the DNA sample or not. How can I figure that out? I can use hybridization. If you remember hybridization, we have discussed about it, where we denature and anneal. We add a complementary strand to it, complementary sequence. This will be 5' to 3'. And the difference is this probe that I'm saying, which is a complementary sequence to the sequence I am looking for, is usually radio labeled with phosphate, okay, radioactive P32. So when we use P32, we will synthesize the probe separately in a different experiment. And once we make that, we will include that they use radioactivity. And then what we will do with the filter is we will hybridize. I mean, before doing that, we have denaturation step, we have fixing step, so that the DNA gets attached to the filter, and then we are denaturing so that the double-stranded DNA will become single-stranded, and then we are adding the probe. The probe will, you will add a lot of enough probe so that anywhere it finds a sequence like this, it will go on base pair with it. So if it base pairs, like this, for example, then that means that the particular sequence I'm looking at is in this band, right? And then you use autoradiography to figure out which of the bands, because we cannot see radioactivity with our naked eye, so we have to use X-ray film and other techniques where you can find out which of the bands actually have that particular sequence. Okay, so if you had sequence sample A, sample B, sample C, and sample D, what can you tell from this? Sample B and sample D have the sequence I'm looking for, that is this one here. And sample A and C do not have the sequence that I'm looking for. So this has many applications actually, and we can always what we can do right now is just understand the principle per se, and when there is as an application, if it is genetic engineering or recombinant DNA technology course, we would learn a lot of techniques, how to use this technique to find out a scientific objective. Okay, that is southern blot. I'll quickly recollect southern blot. It's about invented by Southern. It uses hybridization between the probe and the sample, and the objective is to find if a sequence is present or not, if a particular sequence is present or not in the given sample. It uses blotting, nitrocellulose filter. It starts with DNA restriction digestion of DNA or genome, electrophoresis, then blotting, then fixing, denaturation, and then we have probe or hybridization of the probe, and then autoradiography. Another technique is very similar. It's called as northern blotting. In the previous one, it is about whether a sequence is present in the given DNA sample or not. Here in northern blot, they just put northern to just indicate it's opposite of southern. Opposite is something like whether the objective then is to find if there is a particular RNA or not, RNA sequence or not. Okay, RNA is extracted. You don't need to digest it, or you don't digest it as such. Um, because RNA is transcribed from the genome, it comes as already pre-made smaller fragments, right? mRNA has a length, rRNA has a length, and so on. mRNAs, there are different types of mRNAs. Sorry, different genes are there. Each of them are different lengths, so you'll have different sizes. Then you do electrophoresis, and then we will do blotting, and then we will add the probe. And if the probe is binding to a band, then we would say that the particular sequence is present in that sample, and if it is not there, you can say it is not there. It is typically used to find out what is the amount of transcription of a gene. So if I know a gene and I want to see how much of mRNA is produced, I have to make a probe that is complementary to that mRNA, and then do the hybridization step. Okay, and then we will figure out if the mRNA is present or not, and if it is present, by using semi-quantitative methods, you can find out how much of it is present. Okay, now we will discuss something about PCR technology, polymerase chain reaction. I want to use this opportunity also to revise or kind of review whatever we learned from replication. So it should be two-way thing. In replication, what happens is you have DNTPs, and DNA polymerase will make it into a polynucleotide, right? That is polymerization of DNA. And it requires primers, and the primers are made by primase during replication. And it requires the template. The genome is the template. There are metal ions, Mg2 class, and so on that are required. And DNA polymerase will make polynucleotide. The same thing is also done during polymerase chain reaction. But there will also be several problems associated with that. The first thing is DNA is double-stranded, right? And how does it how does it denature? What is the enzyme? It is helicase. And the denatured one is still is kept in as single strands by single strand binding proteins. And then the primase will synthesize the primers because DNA polymerase cannot initiate replication by itself. It needs this 3' OH onto which more DNTPs are added. So primase does the job of synthesizing primers. And then DNA polymerase will polymerize the DNA. And of course, it requires the template because DNA polymerase is a template dependent um, DNA dependent DNA polymerase, right? DNA dependent, when you say, it means the template should be DNA. So if we were to do in the lab, how can we do that is the question then. So how to denature? One of the ways you can do is using high temperature like this. If you are using high temperature, DNA polymerase will get denatured, right? DNA polymerase is an enzyme. And so scientists have brought in uh, an enzyme called as Taq polymerase, acquired from Thermophilus aquaticus. This should be in italics. This is wrong. The always organism names should be in italics. Anyway, this is Taq polymerase because it is obtained from thermophiles which live in temperatures somewhere around 100 degrees centigrade and so on. That means even if we increase the temperature, they are not going to get denatured. They are very stable at even high temperatures. Then we need to do the job of primase, that is 3' OH. So we can chemically synthesize primers. And here, because should we give RNA primer or DNA primer? If we are chemically giving primers, then why not give DNA? Then what is the what will serve? If we directly use DNA primers, right, then we don't have to use RNase H. I hope you remember the function of RNase H that degrades the RNA part of the RNA primer. If we use primers made up of DNA, then we don't have to go through all those steps, right? With these primers are chemically synthesized, right? In the lab, they can synthesize chemically and then use those as small primers. DNTPs should be provided, and then magnesium ions for the proper functioning of DNA polymerase, and you need a thermocycler. Why do we need a thermocycler? We have to have denaturation first, and then we need to have polymerization after that. Polymerization doesn't occur at the same temperature at what we are doing at uh, polymerization doesn't occur at denaturation temperatures. We will get to that, but I want you to understand about DNA polymerases. We studied about DNA polymerase 1, 2, 3, and so on, and 4 and 5 also. Here we have some of the polymerases that are used in PCR. They are acquired from different types of enzymes. This is the commonly used Taq polymerase, Thermus aquaticus. Um, all I need you to understand is it has 5' to 3' exonuclease activity. And there are other enzymes called as Spirococcus furiosus and so on. They are obtained from those organisms. So this would be PFU. It has 3' to 5' exonuclease activity. Proofreading activity is present. Okay. That means if it made a mistake, it can go back and remove the base that has been added, that is base wrongly added. So proofreading, if I were to clone a large fragment and if I want to polymerize a large fragment, then I better use the enzyme that has proofreading activity, because thermophilus Taq polymerase also makes mistakes, about I think 1 in 10,000 or so. In number can be corrected. 1 in 1,000 or 10,000 bases it makes a mistake. That means if I'm cloning a gene and something I might be getting a mutated one. So if I am very clear about sequence and cloning experiments, I better use something that has a proofreading activity like this. Now we will see what happens during polymerase chain reaction. So first thing is I will try to go along with this. This is the program. Initially, I will write the program here again. So 94 degrees centigrade for 1 to 3 minutes. It depends on the different parameters, but right now you'll just take it as this. This is called as initial denaturation. Then you have denaturation. Step 1, step 2, denaturation, 94 degrees centigrade for, you can see here, that is about 30 seconds. The third step is primer annealing, right? Because we want the primers to be bound to the bases. Let me also try to use this here. Say, for example, the first one would be DNA is something like this, double-stranded and all. And when we initial denaturation will cause majority of the DNA to denature properly. And the next step is denaturation at 94 degrees centigrade. So assume that there was a double-stranded DNA like this, and because of that, it has now denatured and separated into 2 DNA strands like this, single-stranded. I want the primers to bind to that. If I maintain the same 94 degrees centigrade, then the DNA will not bind there. They will not renature. The primer will not renature. So I have to reduce the temperature. And the temperature is reduced to about 55 degrees centigrade. And for say 45 seconds to 1 minute also it can be there. Usually I think in my experiments we put about 30 seconds or so. It depends on the primers and so on. So when we do that, I have to, before I move forward, I have to write this 5 prime, and this is 3 prime. Here is 5 prime and 3 prime. So the DNA primers that we synthesize now should be like this. Why should they be like that, and why shouldn't they be like the green ones that I'm about to draw? Why is that? I'll try to write the primer sequences here, sorry, the ends of these, so you'll probably be able to understand. So I'm having primer 1, primer 2, and so on like that, just for understanding. 1 and 2, green ones, 3 and 4. Which of these pairs is correct? For 1, if I look at, this will be the 3 prime and this will be the 5 prime. And for 2, this will be the 3 prime side and this is 5 prime. If I add DNTPs and DNA polymerase will add, assuming there is more template, it will polymerize on the opposite direction. We don't want that. Our objective is to polymerize this part of the genome that is called as amplicon, the region that we are trying to polymerize. They should be converging. The polymerization should be converging from all these sides like this and like this. And that is why, say, for the 3 and 4, you can see that this is 5 prime end and this is 3 prime end. For the 4th primer, 3 prime end and 5 prime end and 3 prime end. If I add DNA polymerase to a situation like that, then DNA polymerase will add bases to these like this, right? DNA polymerase will synthesize to this way, and this one will synthesize the opposite way. Now, for this amplicon that I'm looking, I initially had one copy. Now I have two copies, right? That is the essence of PCR, where I want to polymerase chain reaction. I will try to describe that, what is meant by that chain reaction. So I hope you understood the primers, where should they be, and how should they be, why should they be like that. So the next step is extension. I already finished that, I think, right? I have drawn these lines here to indicate those are extension step. Now I had one molecule here, which became, now I have three molecules in total. The first one is template. You add a template and then you got two molecules. The same thing, if I repeat 1 second, 72 degrees centigrade extension for 1 minute, it also depends on what is happening. The next step would be, you will tell the PCR machine, you will do these experiments by yourselves, and then you will see that it's just a machine that you have to operate and add some things and know the principle of it while you operate the instrument. So in step 5, you will see that you will say, go to step 2. This is step 2. So you are telling the machine to go and restart from here. Like that, it will keep on going. If you didn't tell how many times, it will keep on going here. It's kind of a loop there. So that means once first step is denaturation, then primer annealing, then extension, and then you're telling it denature again, and again primer binding. So there are certain concentration you add. You will add a lot of primer actually. So you will not be exhausted very soon. Typically, PCR reactions will be made to go for say 25 to 30 cycles. Okay, so the PCR machine will keep on going to these temperatures. 94 degrees centigrade for 30 seconds, then 55, then 72, and will keep on going for about 30 cycles, and then it will stop. Sixth will be, say, stop. Okay, that is the principle, or the core principle of PCR. What I'll do is I will send you some videos on these 3 techniques from YouTube. You can also search and find your own. And probably next time we will do problems. And the problems will be, I think some of you are already doing it well. I've seen some people who have submitted the assignments. We will do restriction mapping. This would be easy if we were in person. Restriction mapping is not easy to discuss on in like this. We have to have individual training, and because different people use different thinking. And there is we will do a problem also on based on PCR, and we will also do based on Mendel's laws. Okay. So if we finished all these, then we have finished the first 2 units of your syllabus. There might be one small topic on ribozymes, which I will discuss at some other point. Okay. Any questions? But otherwise, this is a very fundamental level that we are discussing, because we don't have any course on genetic engineering. I included these topics. And when you are reading your textbook, there are several experiments that are shown. And if you didn't know these things, it would be difficult for you to follow those experiments. I'm hoping that you will read those experiments too. So to give you some kind of kickstart, I added these things in these techniques in your syllabus.